Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
Have you ever looked at those people who seem to function perfectly on just, a few hours of sleep and wondered, what's their secret?
Right. Is it just sheer willpower, or is there something else going on?
Exactly. Like, what if the amount of sleep you really need was actually well written in your genetic code?
That's pretty much the core question behind some really fascinating new science. Researchers have zeroed in on a specific genetic change, a mutation, in people we call natural short sleepers.
Natural short sleepers. Okay, so these are the folks who consistently get, 4 to 6 hours a night?
Yeah, something like that. And crucially, they don't seem to suffer the usual negative effects we associate with, not getting enough sleep. They feel fine.
Okay, so we've really dug into the source material for this deep dive. We're looking at findings from a pretty detailed scientific paper published in the Proceedings of the National Academy of Sciences.
Right, and we've also got insights from a health news article that kind of helps break down this complex research for a wider audience.
Our mission here is to really get under the hood of this natural short sleep thing. What is this specific genetic tweak they found? How does it actually affect the body?
And importantly, what does it tell us about how sleep works or is regulated in, all of us?
So maybe start with the basics. What did scientists already know about these natural short sleepers before this specific study?
Well, they've been curious about them for a while. People with this lifelong pattern of needing less sleep without, any obvious problems. Previous work had pointed to a few genes.
Yes. I think I saw those mentioned, like DEC2, ADRB1.
Exactly, and NPSR1, GRM1. Those were thought to play some role in controlling our sleep patterns. But this new study zoomed in elsewhere.
Okay, where did they focus then?
They looked closely at a type of protein called a protein kinase, specifically one named salt-induced kinase 3. Let's just call it SIK3 for now, though we should probably stick to the full name mostly.
Salt-induced kinase 3. Okay, and what was known about that one before?
It was already on the radar, known to be involved in the processes that govern sleeping and waking. But the key thing is, there wasn't direct evidence linking it specifically to being a natural short sleeper in humans.
So this study was trying to find that direct link.
Precisely. That was the missing piece they were looking for.
So how did they find someone like that? It sounds like a pretty rare trait.
Yeah, they recruited a key participant, a healthy woman, 70 years old, who said she'd only slept about 3 hours a night her whole life, and she was apparently very active too.
Wow, 3 hours. Did they just take her word for it, or?
No, they used objective measures too. They had her wear a wrist actigraph. It's kind of like a fitness tracker, measuring movement to estimate sleep.
And what did the actigraph say? Was it really 3 hours?
Well, that showed an average sleep time closer to 6.3 hours per night.
Okay, that's still definitely on the short side. Much less than average, but quite a bit more than 3.
Exactly. It really highlights that difference between, our subjective feeling about sleep and what's objectively happening.
Right, so they have this individual who demonstrably sleeps less than average and feels fine. What's the next step to find the genetic link?
The next big step was genetics. They did what's called whole exome sequencing on her DNA.
Okay, whole exome sequencing? That means looking at all the protein coding genes, right? Searching for something unusual.
You got it. They scanned all those genes looking for variations, mutations that might explain her unique sleep pattern.
And did they find something specific?
They absolutely did. The analysis flagged a particular point mutation, a tiny change in the gene that provides the instructions for making that salt-induced kinase 3 protein.
And this mutation has a specific name.
Yes, it's called N783Y. That just specifies the exact change, one amino acid swapped for another in the protein chain.
Okay, but finding one mutation in one person, that doesn't automatically prove it's the cause of her short sleep, does it? Correlation isn't causation, as they say.
Excellent point. That's always the challenge. So to confirm it, they needed more evidence. They turned to a mouse model.
The classic mouse model. So they engineered mice to have this exact same mutation.
Precisely. They introduced the same N783Y change into mice and then carefully monitored their sleep. They compared these mice to normal control mice without the mutation.
And did the mutant mice sleep less?
They did. The mice with the N783Y mutation slept on average, about 30 minutes less per day than the control mice.
30 minutes. Okay, maybe that doesn't sound like a huge amount to us, but in mouse sleep terms, that's significant.
Very significant, yes. It provided really strong evidence that this specific genetic change directly causes a reduction in sleep duration. That's a key breakthrough, finding that causal link from a human mutation to sleep amount.
That's pretty amazing. So they found the gene variant, showed it causes shorter sleep in mice. But how? What does this mutation actually do to the protein to make this happen?
Good question. They use computer modeling, computational analysis to figure that out.
Mm.
It seems the N783Y mutation causes slight changes in the 3D shape, the structure of the salt-induced kinase 3 protein.
And that altered shape affects its job.
Exactly. Its main job is transferring phosphate molecules onto other proteins, a process called phosphorylation. Think of it like flipping cellular switches.
Phosphorylation. Adding those phosphate groups turns other proteins on or off, right? It's a key regulatory mechanism.
You've got it. And this mutation seems to impair the protein's ability to do that efficiently. It's less good at adding those phosphate tags.
So a less efficient salt-induced kinase 3 protein means less sleep needed. It's incredible how one tiny molecular change cascades up to affect something as complex as sleep behavior.
It really is. And the fact these findings were published in a major journal like Proceedings of the National Academy of Sciences just underscores how important this is.
Right. It really solidifies the role of this salt-induced kinase 3 as a crucial sleep gene.
Yeah, absolutely. And finding that the effect holds true across species, humans, and mice points to its fundamental importance in biology, something conserved through evolution.
So, stepping back a bit, what are the bigger implications here? Does this understanding just help explain these rare short sleepers? Or could it mean something for, well, the rest of us who maybe wish we needed less sleep?
Oh, it definitely has broader potential implications. By pinpointing these specific genes and the biological pathways, like this phosphorylation pathway.
Researchers might find new targets for therapies.
Exactly. It could pave the way for designing treatments to improve sleep quality, or maybe even help counteract some negative effects of sleep deprivation for everyone. Imagine therapies that could help people feel rested on less sleep when necessary.
That would be revolutionary, though probably complicated.
Very complicated, yes. But this understanding is the first step. It could unlock future treatments for various sleep disorders.
Okay, but let's be balanced. As with any study, there must be limitations, right? Things we need to keep in mind.
Absolutely, that's crucial for good science. The main one here is that the human part really centered on just one individual.
Even though the mouse data strongly backs it up.
Right? The mouse model adds a lot of weight, but it's still just one human case study initially. And while this N783Y mutation was clearly linked to less sleep.
Sleep is complicated. There are probably other genes, environmental factors?
Definitely. The exact mechanisms and how other genes interact with this one, plus lifestyle factors, that's all still being unraveled. And remember the mismatch in sleep reduction.
Oh yeah, the woman reported a huge reduction, like down to 3 hours, while the mice only showed a 30-minute drop.
Exactly. That suggests other factors are likely contributing to her particularly short sleep. It's rarely just one single gene doing everything. Sleep is incredibly complex.
Now, you mentioned this kinase, salt-induced kinase 3, is getting a lot of attention. I think I saw another study mentioned looking at it from a different angle, maybe in the journal Sleep.
Ah, yes. That's interesting too because it provides another perspective on the same protein. That study didn't look at the N783Y mutation associated with less sleep.
It looked at different changes.
Right. It explored two other mutations in the same gene in mice called T221E and SLP. And they were looking at how these affected the sleep-wake cycle and the body's internal clock, the circadian rhythm.
And what did they find there? Did those mutations also cause short sleep?
Actually, the opposite implication in some ways. Their findings suggested that modifying the activity of salt-induced kinase 3 could be relevant for conditions involving too much sleep, like hypersomnia, or problems with waking up properly.
Wow, okay, so one mutation in this gene is linked to needing less sleep, and other changes might be involved in needing more sleep or having disrupted sleep cycles.
Kind of sums it up, yeah. It really paints a picture of this protein as a key regulator, potentially acting like a dial for sleep duration and timing, depending on how its function is altered. It underscores its central, multifaceted role.
So if we pull both of these studies together, the PNAS one on the human short sleeper and the Sleep Journal one on other mutations, what's the synthesis? What's the overall picture of this protein's role?
Well, the PNAS study, the one with the N783Y mutation, found something else quite interesting. It altered the overall pattern of that phosphorylation process we talked about.
The adding of phosphate tags.
Yes, and it particularly affected proteins found at synapses, the connections between brain cells. This hints that the mutation doesn't just change how much you sleep, but maybe also how your brain functions during sleep.
Affecting synaptic processes. That sounds potentially important for memory consolidation or brain cleanup during sleep.
It could be, yes. Their analysis also suggested that this SIK3 mutation might trigger changes in other related kinases, like protein kinase A and mitogen-activated protein kinase. It points towards a whole network of these proteins working together to control sign.
So it's not just one protein, but potentially a cascade or network effect.
That seems likely. And understanding this network is really where the potential lies for developing future treatments for a whole range of sleep issues, from insomnia to hypersomnia.
This has been absolutely fascinating. It really does make you think differently about your own sleep patterns, doesn't it?
It does.
Especially if you're someone who consistently feels fine on, say, 6 hours of sleep when everyone tells you need 8. Maybe it's not just a habit.
Maybe it really is partly down to your genes.
Yeah.
It certainly raises that possibility for some individuals.
And it definitely highlights why scientists keep digging into the genetics of sleep and the roles of these intricate protein kinases.
There's still so much to learn about this fundamental biological process.
So here's something to leave you, our listeners, thinking about. If we continue to unlock these genetic secrets of sleep, and maybe even gain the ability to safely modulate these pathways, what could that mean?
Yeah. What could it mean for human potential, for productivity, maybe even for overall health and well-being?
Could we fine-tune our sleep? Redefine what feeling truly rested means? It's definitely a provocative thought to mull over.
Thanks for tuning in to the Health Headliner podcast by To Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.